In this video, we will discuss about one of the treatment mechanism of Pompe disease, or we can say how to uh, increase the efficiency of treatment mechanism of Pompe disease. So let us first quickly revise what is actually a Pompe disease. This disease is mainly a lysosomal storage disorder, or it is also known as or a disease from a pool of LSD, that is lysosomal storage disorders. This is the image of lysosomes. So what is the main problem in this is that the enzyme, the gene is, that is GAA gene is mutated. Because of this, it is not able to produce the proper alpha glucosidase enzyme, and this enzyme is responsible for lysosomal degradation of this glycogen. So in normal case, when this alpha glucosidase is formed, then it helps in the degradation of glycogen, and that maintains the level of glycogen within limit. However, in case of GAA mutation or POMP affected cells, this GAA is mutated. Because of this, the degradation, this alpha glucosidase is not able to perform its function properly, and the glycogen amount of glycogen with time continues to accumulate within the cells, and with increased amount of glycogen will leads to problems such as weakness. So uh, let us now understand what are the treatment mechanisms that are being designed or focused on. First one, since the GAA gene is mutation, and we know from our several videos that there is a quality control mechanism within the endoplasmic reticulum, and this is represented here as endoplasmic reticulum. So whenever a mutated proteins formed within the endoplasmic reticulum, and since it does not have a exact sequence of the native protein, it is difficult or not able to form a native structure. And when the protein does not able to acquire its native structure, the quality control mechanism of endoplasmic reticulum will either let that protein to continue in the endoplasmic reticulum for extended duration of time, and even after that, if protein not able to acquire the native structure, then it will be directed towards degradation. So this GAA protein in most of the time in case of Pompe disease is directed to the degradation, since it is not able to acquire its native structure. So what approach is suggested in this slide or in this paper is that they will introduce a artificial uh, pharmacological chaperones. So these pharmacological chaperones will help that mutated protein to acquire, to get a improved folding or to acquire a native structure in such a way that it will be passed by the quality control mechanism, or UGGT1 or UGGT2, and then move further towards this Golgi apparatus for further glycosylation or improved glycosylation, we can say that. So when this is passed through the Golgi for the, it will acquire the, or we can say maturation of glycosylation occurs in the Golgi apparatus. Then it will then, since now structure is similar to the native structure, the glycosylation that occurs in the Golgi apparatus is similar to the native glycosylation. Then it can move to the lysosomes, where it can perform its regular function, that is degradation of glycogen. And this will, since the protein has its native structure, the it will improve the it will improve the half lifetime of the recombinant enzyme, or we can say this uh, mutant protein within the lysosomes. With the help of this recombinant enzyme and shap with the help of the combination of let me rephrase the sentence. Since now the recombinant protein has acquired a native structure, and with the help of the pharmacological chaperones, it will be more stable and have a increased half-life period compared to the this mutated GA protein. And another problem that occurs in this mutated GA protein, in case it is able to bypass the quality control of the endoplasmic reticulum, is that because of non-native structure, it will able to get a different glycosylation profile, and this will leads to ineffectiveness or less efficient GAA function within the lysosomes, or in simpler word, defective activities in the lysosome. Whereas the new approach that is suggested, which include the recombinant enzyme, and along with this pharmacological chaperones, will might help or might work towards solving the problem that arises because of mutation of GAA gene. Uh, so I hope you have understood the treatment mechanism and how it will improve the situation in the Pompe disease. We need to understand more about this details, more about this treatment mechanism. You can go through the reference that has been provided in the description and also in this slide. That's all for this video. Thank you for your time.